Hello everyone, a very good morning to all of you and welcome to today's webinar for the day. I am Himashri Bhattacharya and I am a Senior Executive for Science Communications at the DBT Wellcome Trust India Alliance, and I will be your host for today's lecture. Thank you for joining us today. So today we have the sixth and last lecture of the Continue Biotechnology Education Lecture Series, and these lectures were organized by India Alliance in collaboration with Guru Angad Dev Veterinary and Animal Sciences University, in short known as GADVASU, and IIT Roorkee. Our aim throughout the series has been to bring to you a diverse range of topics in biotechnology. So far in the series, we have covered topics such as genetically engineered vaccines, next generation sequencing, metagenomics, genome editing in animals, and structure assisted single molecule multi-targeting of SARS. So today's topic will be on stem cell therapy, and we have with us an esteemed guest whom I'll introduce in a while. So let us before our speaker, let us start with a quick introduction to the organization that I represent today, the DBT Wellcome Trust India Alliance. Many of you may be familiar with the organization. For those of who are new, India Alliance is a charitable trust registered in India that funds research in health and biomedical sciences. So yes, just like the familiar DST, DBT, CSIR, we also fund research in India. But apart from that, we invest in transformative ideas. We support research ecosystems to advance discovery and innovation to improve health and well-being. We are being funded by the Department of Biotechnology, Government of India, and the Wellcome Trust from UK. And apart from this, the funding part, we also support various programs that promote a national and international collaborations, professional skill building workshops, outreach workshops, and also have programs on strengthening the research ecosystem and also promote science education, awareness, and public engagement with science in India. India Alliance also periodically conducts science communication and outreach workshops such as this. This is one of our outreach workshops, as well as public engagement events throughout the year. To keep yourself updated on the fellowships of India Alliance fellowships and grants and other events and workshops that I spoke about, please follow us on social media through the handles that will be soon shown on the screen. So more on India Alliance will be shared with you later during the session by my colleague by my colleague Isha Goel. So before we dive into the lecture, a few housekeeping matters to keep in mind. This webinar is being recorded. Your videos and microphones will remain switched off during the event to avoid any distractions, but you can always leave your questions for the speakers anytime during the session in the question box. You can see a question box which is there, and please make sure that the question only relates to the topic that is being discussed and covered in the webinar today. In case you find that the webinar has ended abruptly due to any technical glitch, please feel free to join back using the same registration link that you have received to join. Please note that we'll be providing e-certificates to those of you who attend the entire session, and after the session share a short post-workshop evaluation with us. These certificates will be mailed to your registered email IDs within 6 to 7 working days. We all look forward to your feedback after the webinar. And if you planned on sharing highlights from the workshop through the social media handles, do feel free to tag us. On the next slide, you will be able to see the agenda for today's workshop. So we begin, as we have already begun, with the introduction. And now we will have, next we will go to a welcome address by Dr. Yashpal Malik, the Dean, College of Animal Biotechnology, GADVASU. Then we'll have our esteemed speaker, Dr. Taru Sharma, Director of NIAB Hyderabad, talk about stem cell research, its therapeutic promises. Each session will be followed by a short question and answer session, during which you can raise your hands. You can see a hand raise icon, and you can ask yourself to be unmuted, and we can, you can ask your questions. Next, we'll have a short screen break, followed by a session on funding opportunities at India Alliance by Isha Goel, Grants Advisor at India Alliance. And we'll then end the session, today's lecture, as well as the entire series with a closing remarks. So I hope you all will be joining us for the entire duration. And without further delay, I will now hand over the screen to Dr. Yashpal Malik. I hope Dr. Malik is here. I think there is some technical issue from his end. So if Dr. Malik is not here, I request Dr. Chaudhary to take it up.
Okay, thank you. It's really an honor to have an eminent speaker, Dr. Taru Sharma, who has been working in the field of stem cells since many decades. We all know her. So biotechnology is an exciting, and ever-expanding field of science that harness cellular and biomolecular process of living beings to develop a usable product for the mankind and society. The very objective of this CBE lecture series, as Himashri said, to strengthen the biotechnological skills and provide updated information of biotechnology to the student, staff, and the professionals, researchers. So on behalf of the College of Animal Biotechnology, GADVASU, IIT Roorkee, and the India Alliance DBT, welcome. I, Dr. Ratan Kumar Chaudhary, welcome you all to the sixth lecture series, which is the last lecture series on stem cell and its therapeutic promises. And today we will hear Dr. Sharma what the stem cell is doing, what it has a therapeutic promises to the animal science and veterinary science. And with that, without much taking time, I think I welcome Madam Sharma to proceed with her talk. So thank you, ma'am. You're welcome.
Thank you very much, Dr. Ratan.
Thank you, Dr. Ratan. So before ma'am starts, I think so many of those who do not know about her, I'll give you a brief introduction to Dr. Sharma. So first of all, thank you, Dr. Chaudhary, for the nice welcome speech. So I'd like to introduce you now to our today's speaker of the day, Dr. Taru Sharma. So Dr. Taru Sharma is the director of the National Institute of Animal Biotechnology based in Hyderabad. She is a well-known, esteemed Indian scientist of high repute. Dr. Sharma's contribution to molecular biology, stem cell physiology, particularly embryo genomics and animal cell therapeutics, is well recognized in India and abroad. She is before NIAB, she was also a principal scientist and head of Physiology and Climatology Division, Director Center for Advanced Faculty Training and Chairperson, and Faculty of Veterinary Physiology at ICAR IVRI Izzatnagar. She has been the recipient of several prestigious awards, a list of whom includes some of the well-known awards such as ICAR Rafi Ahmad Kidwai Award 2018. So I invite Dr. Sharma to now talk about the sound topic today on stem cells and the therapeutics.
I
Over
think
to you,
Dr.
Dr.
Malik is there if he wants to speak
Yes,
something.
just
Thanks,
I will
Hima.
take a minute.
Yeah,
It was
good morning.
really,
Yeah. Good
really
morning.
something with my one of the computer. It has gone out, and sorry for that delay. And really, I am lucky to have Dr. Taru speaking on stem cells. What the long lead she was having on this concept, and I think she is the best contributor in the country, known for
Thank
her
you.
contributions not only in veterinary science, in even medicos or in several committees she is leading at the country level. So we are really lucky, and I'm really thankful to madam for giving us the time when I
Thank you,
talked to
Dr.
her,
Malik.
and she said, okay, I will be there. And unluckily, we have to postpone because of some emergency. But again, this is a good day that we are starting again with that one of the best concept in biotechnology. And purpose of this series was because it might have been narrated that we want to enrich the knowledge, update the knowledge of the youngsters, young minds, and no one else can share the experiences than Dr. Taru. So on my behalf and behalf of our university and IIT Roorkee, who are our partners, really we are thankful to you, ma'am, for sparing your time.
Thank
Please,
you so much.
please.
Thank you for being very kind, Dr. Malik. Thanks, Hima, for this. So, may I share my screen, Hima, if you're good?
Yes, yes.
Okay.
Hima, some people are not able to join. Can I get a link so that I can share them
Yeah,
personally?
even here, I also got some such.
Yeah, I'm
Okay,
getting
fine.
a message.
For me also
Okay.
there was a issue. I shifted to
Ha, ha,
another.
ha, ha. So, I believe my slides are seen.
Yes,
Yes, ma'am.
visible.
Yes. So, may I go on, Hima,
You
or
can
you want me
start
to wait?
now.
Yes. So it is
Thank you.
9:44. You said that I can take 1 hour, so I will respect that time. Well, it's such a wonderful morning. It's not that because I'm talking on the area wherein we have been working last couple of years, more than 1.5 decades. But also, it's a wonderful opportunity to get linked with not only the seniors, the researchers, but also the upcoming and budding students through this Biotechnology Education Lecture Series. It's much appreciated a series. I myself follow it through DBT Wellcome Trust YouTube link. And I have all applauds and appreciations for that. And I feel pretty fortunate. Credit goes to my good friend Dr. Malik, the Dean of School of Biotechnology at GADVASU, one of the very prestigious veterinary university. Rather, if I say it's one of the most happening university in the country in the veterinary sciences. And I personally know not only him, I personally know a couple of other colleagues because we do collaborate through One Health Consortia. And also a couple of my own students who are now a part of this vet school. It's there. Joining them was IIT Roorkee, again, very dear to us because we have a couple of good friends with whom we do work and we do collaborate. So thank you very much for that. I come from DBT NIAB, as Hema already said. So what I'll do is maybe I'll take a minute or so to share with it that what my institute is doing. My institute is located in Hyderabad, at Gachibowli, and our motto is very clear. We do anything, everything related to animal health. And our motto is ultimately it should go for the human welfare. This is the institute. It's a pretty unique institute in the country, which is focusing only on animal biotechnology. And it was a vision of Professor Ban 10 years ago. So we do have a large and small livestock with us, say caprine and bovine, and our small animal house is very, very good. Besides other things, we do have a vaccine testing facility which was established 2021 when the COVID was really there and everyone was, so we did join hands through PM Care. What do? We do a couple of things. So anybody can visit the website, but just to let all of you know, these are the major areas wherein we work. In the infectious biology, we do take care through bacteria, virus, and parasitic infestations, and also do the adjuvant and delivery system. Our team reproductive biotechnology is very strong, wherein we are focusing on various aspects which are related to production and health, and so is the transgenic technology, therapeutics, and stem cell therapeutics. We are further enriching, and we are also focusing our through our nanobiology group, wherein we are trying to generate the organoids. Different pipelines and omic analysis and molecular docking work is being taken care by the bioinformatics team. And we do take care of a nutrition part as well, especially the human gut microbiota is pretty amazing. So one need to explore it, and that's what we do. Just to tell that we have a couple of important flagship programs. This is a pride of NIAB when last year the group bioinformatics group led by the former director has generated the global high density SNP chip and India's first, of course. So right now what we are doing, utilizing this HD chip, we are trying to work with NDDB to associate certain phenotypic characters. We do have another important flagship program wherein we focus on one viral, one bacterial diseases, and they are important zoonotic diseases. And we have important leads from both, especially if I can highlight for JV, against the NS1 protein of this pathogen, we have an antibody, and the detection kit, the LFA electrochemical method has been used. It's very much patent protected, and we are ready to transfer. In the beginning, when the COVID was in air, and before that itself, the DBT conceptualized having One Health Consortia and zoonosis and One Health Consortia. It has equal number of medical and veterinary partners, and including the Northeast partners in a wildlife centers, because without that we are incomplete. Now I come to the task assigned to me. So I'm supposed to speak on therapeutic promises which are there through the stem cell research. I will take you all through, considering the heterogeneous group, having very, very esteemed people who are also working on the stem cell area, and some of the very senior people, some of the younger ones, some mid-career, and the students. So my talk will take you from a very simple, very, very simple introduction towards the data which we have generated, and also the data which is existing in the literature, and where are we, and what are the promises. And for sure, since I work in an Institute of Animal Biotechnology, so my talk would focus on animal aspect. Before this, as was told, I was at ICR, Indian Veterinary Research Institute, and a good amount of data what I'm going to share was generated there with the team, interdisciplinary team over there. However, I would like to touch upon what one can think of doing in the human beings as well. So the first, let us interrogate ourselves. Why exactly we are focusing on this? It is hardly late 90s or early 2000 when the concept of stem cell biology came. Well, being a physiologist, the very simple thing which comes to my mind is that it's the idea of shifting the basic physiological paradigm towards the therapeutic interventions, utilizing such robust points which comes out through stem cell research. What are they? Let's give half a minute to understand why are they so special. These three things make them so special because they do divide and continuously renew themselves. for a longer period of time. And we all know that though they are specialized cell types, but they remain unspecialized into their niche. And we also understand that they differ themselves from other kind of cells. And why so? Because all the differentiated organs, they do have an undifferentiated niche where these stem cells are sitting. So the meaning to say whichever organ it could be, these stem cells can replace the diseased or the damaged cells, which are not going to heal or renew themselves. The uniqueness of stem cell, as I elaborated, so they do differentiate themselves and they do self-renew for a longer period of time. For understanding of taking you along with me, I have made it a very simple, these lines. Let us classify them. We can do it based on the origin. From where do they come? If they come from embryo, we call embryonic. If they come from adult tissue, adult. And from the fetal tissue, fetal. Very simple. What other things could be? But there are, it's not as simple. You might have been hearing all these kind of names. Let's understand from the embryonic, if they come from very early stage of embryo, say before 8th cell stage, we do call them totipotent stem cells. And that's how I wrote zygotes. We call them pluripotent embryonic stem cells when they come from late morula or early blastocyst. And likewise, multipotents, they are the adult ones. Fetal stem cells, though they come from fetal origin, but they are also termed as multipotent mesenchymal stem cells. All right. So let's see the pyramid. One thing through this pyramid is all of us understand the Maslow's model, and whatever occupies the base is the dominant one, and surely the embryonic stem cells are the one. Amniotic or the fetal ones, they are not in the base. However, they make themselves in the category of the multipotent. However, they placental and amniotic as well as umbilical, they occupy a very important position. Please remember this because I'm going to talk something else. And the adult stem cells, which eventually I'll take you along. Okay, let's understand for why all the three layers, endoderm, ectoderm, and mesoderm. So what data I'm going to share is mainly going to be the marrow origin cells, which we surely call mesenchymal stem cell, and they are adult stem cell. Else, they'll be of fetal origin cells, but they will also be termed into the multipotent cells. It's not that simple as for meiosis, mitosis, but they have their own cell division type. They follow both symmetric as well as asymmetric. And this picture is very simple. And we have the stem cells out here. So they divide through symmetric followed by an unsymmetric cell division and end up getting a progenitor cells. Once we have the progenitor cells, they would get terminally differentiated and you have got a differentiated cells. It could be anything with which we are interested. I got this table. It's more or less a repeat, but what I want to emphasize through this one is that why are we so much talking about this adult, embryonic, and the pyramid and all? Because my talk is mainly on the therapeutic promises. So the take would be which are those cells which we take along with us and move ahead with the therapeutic applications. So do embryonic stem cells make a better options or do adult stem cells make a better option? For sure, for clinical applications, adult stem cells make a better option. However, we cannot leave this very important group of cells, which we are terming as embryonic stem cells. While I move with the details, I'm sure you will be able to understand why do I say so. Okay. Before I go and explain about different kind of these cells, let me just impress upon you what kind of work last 18 years or so we have been following, and what did it lead to, and why is that so important, and where are we? So one is that our major emphasis is on two important aspects. One, on an orthopedic side, we take a bone and bone repair, and on the surface side, we work on a wound healing. So this is one using different scaffolds. That also I will elaborate, but right now I keep it simple. So we do the bone repair and the wound healing. As I said, I will be sharing further more detail and make it very simple for you. Also, we work on nerve and cartilage repair, work through different mitogens and do different things utilizing the adult stem cells or the fetal stem cells. But what is that which ensures that the cells which you're putting over here are doing their job very well? So the important part is one needs to really track these cells in a live model, so in vivo model. So we do track cells, and that's what right from the day we started tracking them, it made us a little more knowledgeable than before, and we could really get a clear picture as what these cells do, how do they move, and it's very easy for us to converse with the owners of the patients, because in case of animal veterinary sciences, the patients are not talking to you, so you are approaching through owners. Very important part which I like to highlight, especially the diabetic wounds, and some of the work which we did using the camel milk and diabetic wound and its repair, and also along with the stem cells. So the work which is related to embryonic stem cell, is it important? Where does it lead? What does it tell us? That makes also a sense. So I'll give a brief touch on the embryonic stem cells of the buffalo region, because that's what our lab could produce. And I'll also give you the fragrance, what other embryonic stem cells, including induced pluripotent stem cells, do, and what one can think of doing with that. All right. So what we have been doing, the team at IVRI was fantastic. My colleague and my strong collaborator, surgeon, Dr. Amarpal, and his team was working with me. And these are some of the OPD pictures wherein we were focusing on spinal cord injury and paraplegia cases, which are really very difficult to treat. So what I'm just showing here, the compression in the thoracolumbar region, and exactly the spinal cord injury and repair. I will share the data, but I'm just showing here, this has been exactly we have been doing. And once we understood the whole mechanism, then we did a very large study utilizing 68 patients and giving the stem cell and doing a lot of things. This I will share when I share the data. This is one picture to impress upon what two of us, what his team and my team together could address was bone defect for sure, as I said, wound, as I said, and the spinal injury. These are some of those patients whom we could really treat and could really understand what good these cells can do and what promises it hold. Let me take you to embryonic stem cells first, very quickly. And why do I talk about it? The list is very long. In fact, I kept the list short. It's longer than this. In case of farm animals, or in case of animals, not only farm animals, pets also, equines also, one can think of utilizing embryonic stem cells, which we term as pluripotent stem cells. One can think of doing all these things. All right. The take is on a transgenesis and cloning. Ever since embryonic stem cells have been understood by us, the somatic cell cloning has taken a back seat compared to the embryonic stem cells. So, what one can think of doing, you have to generate the cells first. And these cells are non-adherent to plastic. So, you need to generate a feeder system first, or the synthetic matrices first. One of the students who was doing all this very intelligently is part of Dr. Malik's team in physiology division. And we have been using very many growth factors. One thing is that these cells are LIF and FGF dependent. All the work has been done and shown. So this is a glimpse of some of the data which we could generate. These are the buffalo embryos, beautiful blastocyst. And what you need to do is, as I said, they are non-adherent. You need to generate the feeder. So feeder also has to be from those cells which hold high telomerase activity. And then the inner cell mass you can either place and generate the embryonic stem cells, or you can isolate and put the whole blastocyst without zona pellucida after hatching, and you get the embryonic stem cells. What do they have? This is the one, and these are the surface markers which you really understand, OX, OXN, and OC. So if they express it, you say so that yes, they are. These are the cells which eventually can differentiate it to other cell types, epithelial, neuronal, fibroblast, and can generate embryonic bodies. For sure, they have high, the telomere length is more, and they hold high telomerase activity as well. And this is a karyotyping picture typically for that pattern. It shows the most important part is that these embryonic stem cells are, you remember I said that they are not as good for therapy as the adult cells, and why so? Because they are teratogenic cells, they form teratoma. But how do you prove it? So this is a paper wherein we could really insert the embryonic stem cell. You need to use a skid mice, and it forms a teratoma, and further you work out through all the ecto and the meso and the markers and say yes, they are teratogenic cells. This was a paper from IIT, got published in 2012. Till date is the only global paper which really highlights the limitation of using these embryonic stem cells for therapeutic as they are teratogenic cells. We further went and utilized the embryonic bodies, formed the different experimental designs, and again inserted these ones into the skid mice, made a picture of all endoecto and mesoderm, and put a markers over there and did a whole immunohistochemistry, and then we proved and also we expressed them. So yes, they are teratogenic. So what's the point we are making, or what's the point which is being understood by us is that you need to really look for some other cells, else you give little more time to get them differentiated. Okay, I'll take you along with me. It's not that what I just talked about, the bone wound and doing the cloning and transgenesis. No, the list is long, and in animal veterinary science sector and in human too. I would like to emphasize one thing, that though we can think of working on a mouse and a rat model, however, the large animals, they do make a gold standard for anything, everything which is going to get translated towards a human science. So one can think of remodeling and tissue engineering. One can also think of understanding the early embryo development, and so much the drug testing and endangered species conservation. This. Moving ahead, the year came 2012, when everyone started talking about induced pluripotent stem cell. And why this year is so remarkable, because it was Shinya Yamanaka and Sir John Gurdon. They received a Nobel Prize for they gave the Yamanaka factors, and these are the 4 Yamanaka factors to generate the induced pluripotent stem cells. Interestingly, the year Shinya was born, Sir John Gurdon gave the concept the genes are not lost and you can anytime reprogram, and he did prove it. So what do you do, and why are they so special? They have the similar properties like embryonic stem cells, but they come out from the reprogramming of the adult fibroblastic cells by inserting the Yamanaka factors, and once you have them, these are induced pluripotent stem cells, iPSCs we call, and it can give rise to all different kind of lineages, what probably embryonic stem cells give. Okay. A historical timeline Oxford University Press gave, and you know, they are yet to do it after 2017, but or we can ask. The important part I like to show you, it was 1998 when the derivation of first human embryonic stem cell line came. It was Jamie Thompson. Jamie was, when he was doing it, well, he was sitting in a human hospital and he did it. But basically, he's a veterinary pathologist, and he has, the credit goes to him. Afterwards, he did work on the human iPSCs as well. Took it ahead for clinical trials too. And this was a Shinya's group, and this Takahashi and Shinya, they gave a lot of outputs to our understanding, and things started moving very fine. Okay. One can think of, this is all I'm talking in case of human sciences. One can think of doing it in animal sciences also, but that would be a longer path, and we really don't want to do that. But one can really think of doing anything which I have mentioned here, and other than those things which are not mentioned over here. Most important is especially deriving the cardiomyocytes, going for the cell therapy, cell-free therapy, utilizing exosomes and microRNAs, going for the drug testing, and one could think of doing not only the disease modeling, but also the bioengineering, the tissue biology. There are limitations as well, and if the picture is good, the limitations as well. All right, I'll take you ahead now towards a major topic of therapeutic application. When we think about, I did not write veterinary sciences or human sciences. This is in general, anybody, everybody is trying to do that. Of course, rheumatic diseases and some of the anti-aging diseases and arthritis is common in animals too. Okay, so these are all the bubbles which really make us understand that we can think of. I will really take you along further with the data which has been generated on the cell-based therapies, of course, stem cell-based therapies and its products. And what are those products? So I will detail it out, what exactly it is. A big review we wrote, and we told that what could be the importance if we move from basic biology to its clinical intervention. And here comes a picture of mesenchymal stem cells, or what I was telling in the beginning, adult stem cells. So, you know, these are the cells, you name it, dental pulp, dermis, cardiomyocytes, endometrium, and so on and so forth. You get MSCs from this. Okay. You get neuronal stem cells also. They are also MSCs, but you can generate it. My data is mainly either from the marrow fluid or from the fetal origin stem cells. The embryonic one which I showed was just a glimpse to make you understand that they are teratogenic, and unless you differentiate, you will not take chance to take it ahead for the therapy. What makes these cells so very important? Number one, they do have a differentiation potential like other cells, like other stem cells. They do have a homing property and they migrate themselves. Else they allow the niche cells to do it. The most important is not what they secrete, the important growth factors, matogenic factors. Important is they are pretty much immune compromised cells, and that's make the task much simpler when you talk about the therapeutic process. All right. Let's talk about a bit of a clinical applications. So just read the two bubbles, and I will focus on these 3 points which I just mentioned over here. How do they evade this allergenic rejection once given? So as I said, they are hyperimmunogenic. Anything has to be done with the expression of MHC class 2 and class 1 molecules. So MHC class 2 molecules are not expressed, and class 1 molecules are expressed very favorably, and they do modulate the T cells phenotype and also create a very robust immunosuppressive local medium. So the task is almost, we are almost at it. So yes, we move ahead with these cells. I'll just give a glimpse of it, what exactly these therapeutic applications are currently being taken in the humans. 2 days back, when I was talking to a group of people at our Brain Research Center of DBT Institute, they put a question, so I inserted it over here. Yeah, our medical colleagues, they are using the marrow fluids and marrow transplants. They are very, very successful with leukemia, lymphoma, so many other things. Umbilical cord blood is also one very rich source of the stem cells, and people are thinking, not practicing as much, are thinking as an alternative of a marrow transplant. And there are some other disease things which people are thinking. Sorry. What could be the 6 stages? I got this picture for my talk, which is a very simple thing. It's not as simple as I'm showing, but one can, for the sake of understanding, if you have the stem cells and then you just collect it, and then you have to have the frozen stem cell bank with you, especially in case of animal veterinary sciences. Your stem cell bank has to be very rich, because patient's owner won't come as per your requirements of availability of the cells. But the similar picture is true for medical sciences as well. And then this is the way they go ahead, and body recovers. And we have some very, very personal close examples wherein people have gone through this therapy, the leukemia patients at AIMS, and it's doable. Let me talk about a little bit of basic biology of these stem cells. As what do they do? As I told you, they do have a homing property, so they do migrate towards the site of injury. They themselves secrete this local robust immunosuppressive milieu, and also they get differentiated. They also have the anti-apoptotic impact, and besides the cells, they also pinch the sitting niche area cells to do all that. So meaning to say, it's all happening in a cumulative manner. This cartoon we generated for ourselves, I keep showing it many places. It's very simple. What it is when you really transplant, all the 3 things will happen for sure. They do differentiate, they do the immune suppressive activity, and they do secrete a battery of mitogens which keep on doing all this function. And okay, here when we say they do secrete a lot of biomolecules, a lot of growth factors having very important property which are required for the required clinical conditions. So the condition media, what is condition media? I'll talk about it. So in my talk, I will talk about the condition media obtained through the stem cells. It's a secrete actually, so it has many trophic factors, and effect of this on a therapeutic potential and its use. Then I will take you towards the use of stem cells, and then I will talk a bit on exosomes and some of the scaffolds before I complete my talk. Let's talk a bit about the condition media. Okay, how our story started, I like to share in just 10 seconds. It was way back when we were, one of my colleague, as I said, Dr. Amarpal, the student was struggling to generate MSCs. So we generated, and he did a radius is the bone of interest which we usually focus. All our SOPs are developed using mainly the radius bone. Doesn't mean we do not repair others, but mainly radius. And this was an allogenic therapy, and it gave us a lead of 1 complete year. The X-rays are pretty much clear. So from there we started, and then we moved to wounds, as I say, and after the marrow origin MSCs, we started generating fetal adnexa MSCs. I'll share a detail of therapy detail of these cells. And then this picture, this is showing that what all you get, what are the important growth factors which are secreted out of it, and why should you use it. And of course, we do track cells, any cells in every cell, and we do maintain stem cell repositories, the frozen ones, and of late we have been scaffolding, and this is very important. Not only we do the biomesh scaffolding, we have been using a periosteum, but here the procedure was a bit longer. One has to decellularize the first and then refurbish the stem cells into it. Of late, we have been using the the amniotic origin scaffold, and this I will touch upon a bit during the talk. Quickly, what is wound healing? Yes, this is what it is. All the 3 points very simply, but it's not so simple. If we see the important phases like coagulation of the blood, inflammation, and addressing the proliferation and remodeling. So what exactly the tissue repair using the stem cell does, it gives us a scarless wound. And why so? Because it has those factors, anti-scarring factors being produced by them. And when we were doing all this work, a very interesting paper came in 2015, wherein they were using surgical meshes and they were coating it with MSCs. This gave us an idea when they are doing it in human being. If we start, enriching the meshes with our kind of stem cells, what will it do? So we started from that time onwards. But I'll focus on here. As I said, they play an important role, and they do modulate in the response of chemotactic signals by repairing and facilitating the tissue regeneration in the damaged tissue area. And these cells, as I said, that they do have a high degree of flexibility, and that is how they are capable of regeneration of skin progenitor cells when we talk about the food. Let me move ahead. This paper of Cell Biology International 2013, very good paper, International Federation of Cell Biology highlighted. It's a very dear table to me, So what it gives all these things, and what all it does, see what you want. Be it a bone repair, be it a wound repair, cartilage, anything. Such a nice table being generated by this team. And then they gave a cartoon also, which is making us very clear. It's like a filter, You just put the cocktail, the wonder cocktail, it goes, and as I said, the cell has a homing mechanisms. So during the injury, the niche cells already are doing the homing, and you are facilitating it further. Keeping these two things in mind, I move further, and I just say over here, not only we have this paper, so for tissue repair and regeneration, there was a paper in 2003 we have done using the diabetic rat wound healing model, only using this conditioned media, and after that, a lot more data. So important points are these 4. Angiogenic, VEGF, TGF are there, mitogenic, anti-apoptotic, as I already told, and for sure anti-scarring. In case if there are some people who are from the medical background, and if not, then also in animals also, especially pets, we don't want any scarring, and we want a better repaired wound when it comes. So this is all what this wonderful it contains, and we exploit it. This is another study wherein we have done it very systematically. I'm just giving a little, showing you a tip of an iceberg. It's a guinea pig model wherein we repaired. So if you get advantage of a week or 10 days, it's pretty much advantage. And it's not a question of days. It's a question of the kind of hair growth later you get and the kind of wound you get repaired. So further highlighting through a lot of histopathological data and other details, yes, it's very doable. So how we concluded it for ourselves, and this is all which has come out through our work. It has all the list which is shown over here, and the paper which we published in 2003. After that, a very detailed work we did for just because of the alternative of stem cell therapy. I'm sure many of you might be thinking and having a question to yourself that once you talk about the stem cell therapy, what's the need to have an alternative of that? Use stem cell directly, but there is a reason. Well, ICMR and DBT did generate the stem cell guidelines for the use of human stem cells, and from 2014 onwards, we have been struggling, and we still are not having the guidelines at place. However, well, I didn't leave the thread, and I'm still working at it. And soon, if all goes fine, we should have the guidelines. So, but still, I'm sorry. I'm sorry. It's a typical situation of blessing in disguise. So you have a very robust data, and what could have been trash otherwise, we could really generate data. So it's also the concept what government of India is focusing from waste to wealth. Yes, had this been not been there, we would have been trashing it, which now we are not doing. I'm very pleased to share that IVRI has could release our technology through ICR in May 2022, and it's ready for commercialization with Agree Innovate, wherein we are using it in a lyophilized mode with differentiation. It's not being stopped there. Now we are inserting the various other cytokines and exosomes and mRNA, and we're cross-linking and doing other work and making a different formulations if we can take it ahead, which eventually we should be taking because it's our duty. Now from conditioned media, I switch and show you a glimpse of those stem cells. I showed the stem cells which are of the marrow region. Now I'll just share a small work, what people can think of generating the cells, anything from the fetal net cell, or else you can focus only on the amniotic membrane and generate these kind of cells, simple implant cultures or the routine the stem cell isolation way. You can get them differentiated, try lineage differentiation to prove they're the stem cells, do the ICCs and get all the indigenous surface markers done, generate the population doubling time. Less is the population doubling time, more is better are the cells. And this is what is this particular bar says. So for sure, the water and jelly was the choice of the cells out of all the 4 fetal nets. And we religiously do that. Why waste time? And data is enough. And that data was not only to generate cells and show, it was post-freezing, cryopreservation, and then utilizing fresh and frozen cells for various wound healing. So this is a one wherein we are saying PT is a post-thawed one. So these are the post-thawed pictures, and what I really feel, with all humility, I share this work is the one which he published in 2016 and 2017 is the benchmark for us, and more majority of the protocols we just repeat to do the more important work towards the therapeutic use of these cells. As I said, that water and jelly did the best, but others were doing fairly better compared to the control. So now what I do is I also show you the picture and let you understand that should you take the marrow origin fluid, because collection of marrow fluid is surely very painful. You go through iliac crust, use a different needles, and then you collect the marrow fluid. Be it even animals or human beings, it's very painful. So if you can get it from the fetal origin, should you not compare to say which one is better? And we did so. And a very worthy student of mine who worked later with the same Civilier stem cell group, Ratish, did generate all the data. And he said so that you can use either of them with equal, depth of the clinical understanding. I like to share one more work. I clearly have 20 minutes with me, so I have time. So this work, why I'm sharing is, if you remember, I did talk about that one can think of differentiating these cells, and they themselves get differentiated once you put them inside, post homing. So the question we rose to ourselves, if you recall, I was talking about the expression of MSC, expression or no expression of MSC class 2 molecules onto these cells, and yes or no, we knew that if we use undifferentiated cells, they are immune compromised cells, and we are very safe. So we used those cells, but we raised a question to ourselves: should we get them differentiated to get a better picture? For example, if they get to osteogenic, adipogenic, and the third lineage, so shall we be using osteogenic lineage differentiated cells? Or shall we be using chondrogenic lineage cells when we go for cartilage? Yes or no? So the question to us was, do the class 2 molecules get expressed when they get differentiated, or the picture remains same? So we really struggled to search the literature, and we could not find much. So we made ourselves a whole experiment to it. If I can talk in a summary, we used 3 models. We used rat, guinea pig, and rabbit model. The data I'm sharing here is a guinea pig, but of course the rabbit model gave us a very clear and a better the bone details compared to the guinea pig. But guinea pig we use first before using a rabbit, and this data is of a guinea pig. So what we did is, and these are the surgical procedure, 5 mm bone defect. And these are the pictures which are showing one has to use hydroxyapatite or laminin or some gelation so that your cells are housed when you're going for the bone repair. And what we really understood after this work, it is published last year, the tissue is said. And what we said after it, you may go for differentiation, no problems, but you can very well use undifferentiated cells. So what is the importance of this work? Importance of this work is people who work on this, my own friend, Dr. Ratan, who works on a mastitis and a use of mammary gland stem cells, he can well understand that if you go for differentiation, it's a 3 weeks task. So 3 weeks are with you. That's an important outcome of this work, besides understanding the biology behind the differentiation. All right. I take you ahead. Earlier, what we used to do is, we used to do in vivo tracking efficiency of these MSCs, but through the wound pinching. It was the DNA labeled dye from Sigma we were using, and we used to pinch the wounds on a periodic intervals, and we did say that, yeah, this is the tracking. But we understood something, but it was not enough for us. So we decided, no, we will do the in vivo imaging. But imager was not with us at that time. And now it's there, of course. NIAB has a very, very good imager. And now IIT also houses. So we generated a very big study. The animal study, as I was just sharing with you using the canine patients very systematically for the spinal injury work. But for the spine, before giving the spinal injury work to them, what we did is we generated the luciferase. Luciferase is an enzyme not present in the immune system. So these are the two types of data. And these are our transfected cells post antibiotic selection, one can appreciate. And these are the results of our luciferase. Say the bar is taller enough for you to understand. And of course, these are the cells which we could generate and differentiate it. So these are the tube control and the ones which we were having the transfected ones, and the in vivo tracking work. On every 6-hourly basis, we could understand that what does it do. As I said, we use this picture, remember I showed when I was saying I'll detail it out. So to detail it out for you and keeping the story short, what we did, we have taken, it was actually a PhD of mine, Irfan, and a PhD of Dr. Amarpal, Shiva. These two boys, along with a big group, they worked together. Well, as long as cell generation and making a, the injection and doing everything with cell, it was our baby. And when it comes on a clinics and generating the x-rays and all, it was the clinicians who were doing it for us. And that was a wonderful marriage of convenience to do this work. So what we did is we generated all the x-rays and understood a different type of spinal injury. We took different kind of animals. We divided them into small breeds, large breeds, pet animals, and a shepherding and the guard dogs. And then we used them according to body weight. And then we said female, male, all that. A big thing we did. And after all that, what we have done is here we not only use the stem cell, we also use a conditioned media. And this all we said, and this paper we published in 2018, a very important paper using 68 canine patients for spinal cord injury of MSCs in an allogenic mode. So friends, the point I'm trying to emphasize is therapeutic promises are not mere promises. These are the realities which are existing, and the data is strong and clear for our understanding. Yes, it's doable, and more people need to join hands to do it. Coming to scaffold, I'll take maybe around 5 minutes or so, for maybe 4 minutes or so for scaffolding. So generation of this augmented scaffolds, because I used to have a very weird thought, and when there is a handy plus available for human being, why the stores should not have some scaffolds wherein as a owner of pet, we have been pet lovers, always have had right now the Oscar the Beagle, previously the Doberman. So the problems we faced, and like many pet lovers would face. So unless you scaffold, it will not suffice the purpose. So with that thought, we moved on, and what we did is, we generated the cells. Of course, this is the marrow region, as I said, from the iliac crest, and then we did also face antigen, and then we differentiated. This is the work of using the marrow region cells, not the fetal. Generated the stem cell enriched scaffolds. And what did we do after that? We did a lot of things, but just to show you a little bit, use them for wound healing. What does it do? And after that, we understood through that data that yes, this is the way one should really move ahead. In the beginning, what we were doing, we were using only the gel scaffolding, right, for the osteochondral and the wound healing repair. But then we started using scaffolds, and this is guinea pig model work, which I'm showing here. I'm showing a paper which was published last year. And for this, it's a patent protected one. We moved ahead. This is the work, one other laboratory which works with my laboratory is generating a nanofibers, electrospin nanofibers, and scaffolding it with the stem cells. This is the work which I shared, which is published, and unpublished part is patent protected. And this is the periosteum work which I was sharing. It's not now periosteum. Now we work with the amniotic membrane as well and the nanofiber scaffold. So focus is right now with other approaches. Scaffolding and taking it ahead is a better approach other than the lyophilized conditioned media and exosomes and so on. We did not only scaffold or did the bone and wound repair. Simultaneously, we dug deep to the molecular and signaling pathway as well. And we gave a task to a very worthy student who she worked on cell signaling, stem cell signaling pathway. So it's pretty cumbersome, you know, independent ones or dependent ones, as we all know, into any of the cell signaling work. And similarly here. So we chose RUNX2 as a master molecule and try to understand what exactly is happening. And we focused on osteogenic pathway, not on chondrogenic and adipogenic, because this was the one we, as I told you, that we were focusing on bone repair model and wound. So we worked on that, and she did share the data and highlighted. It was a siRNA approach through which we were trying to unveil the signaling pathways of, this is a canine origin cells, I must emphasize. And what we said that the impact is there. The gamma secretase is the one along with SMAD4, which gives a knockdown impact on the osteogenic differentiation, which is being done via RANX2 for the canine origin cells. But this is only half story. Then one INSA fellow joined with me, Ayan Mukherjee, with veterinary biotechnologist from West Bengal University, Kolkata, along with the one of the staff in my lab, Swati. These two, they generated a data for adipogenic and osteogenic further. And right now what we are doing is we are focusing on LOX molecule, which Swati is working. And Ayan is still trying to unveil the osteogenic. So what I'm trying to say that simultaneously we need to understand the basic biology, cellular molecular at cellular molecular level. So we are very much confident and conversant with whatever therapeutic applications we move on. This is the work we started at IVRI and elaborating it over here, not only focusing on, this is a temp picture of exosomes, and we are right now going with the RNA work also, and further exploring it out. How can we isolate exosomes and use them for scaffolding, either separately or along with the conditioned media, lyophilized or fresh. Okay, so much is written here, don't bother what is written. I will explain and make you understand why do I write it so much. See, all the veterinarians sitting in this group who are with us, they understand how much is a long bone fracture a trouble, especially in livestock. Equally important for equines and pets also. But in case of large animals, well, the worthy surgeons across the country, they could generate lot of patent protected pinning, nailing, and plating technologies for the long bone fracture. And of late, last month itself, the same group with which we are working, they have generated a bigger study for this long bone fracture. However, the severity is much more and management is beyond thinking, if I could explain. So the bone tissue engineering does have larger challenges, and I'm sorry, if the tissue engineering, inserting the stem cells, scaffolding them towards a proper direction is very much the need of an hour. And if 10 patients are addressed out of 40, still the agony of owners and before owners, the patients could really be addressed. And we are seriously at it along with the colleagues. Now we signed an MOU from PVR, Telangana Veterinary University, and other colleagues from IVRI. I continue working with them, and some of the CSIR laboratories and IIT laboratories are hand-holding with us for scaffolding, be it a hard titanic or titanium implants, and be it a soft, which we ourselves are generating. And my own scientist colleague at NIAB are helping through the nanofiber scaffold generation. So this is a picture which the first one is showing, which I more or less explained it to you. The second one is which is ongoing, and the third one is we have just started. Last year, I could select two scientists who are making a 3D models for us, bioprinting and biofabricating, and another scientist who are very rich in organoid making comes from Israel. So we would be able to move ahead with further strong ideas what we have had. So what are we really doing at NIAB now? As I told, electrospinning various nanofibers, the list is long, and doing a different 3D printed scaffolding for as a bone scaffold for the segmental bone defect, and doing it in the different composition. People who are familiar with sol gel conversation, they understand. Doing a bioprinting, 3D bioprinting with required mitogen, growth factors, finishing media, exosomes, lyophilized stuff, and the list is long. And this is what exactly our aim with which we are moving. Two animals we are focusing, rabbits to generate proof of concept before we move to caprine, and then eventually with OPD to canines, which I am still making a repo because way back at IVRI, way back in 2021 at IVRI, OPD patients did believe us, and we have had a repo. It takes time to generate a repo with them. However, we will be doing a biofabrication and organoid making over here. This is the point, what I narrated. And with all humility, DBT is kind enough to have trust in me. And I'm sitting right now with the third draft of making animal stem cell guidelines countrywide. Presentations are over. Brainstorm is going on, and soon I need to have best wishes from all of you. Maybe I can request some of the colleagues who work on stem cells, especially animal veterinary sector, to join hands to make all these things happen and let it be there at place. So it will help us commercializing not only the stem cell alternative, but also the stem cell itself, and will not only depend on exosomes and conditioned media derived from cells. This is stem cell team NIAB. The students, or what these students were doing. Right now, Ananya is focusing on a signaling work along with Swati. She's right here. Dr. Sandeep Goyal has his own laboratory, and he focuses on adipose derived stem cells. Sunu works on generation of any kind, every kind of nano scaffolds. We know there's the one who's making all sorts of organoids for us, and Sonu is also generating biosensor platforms for detection of differentiation potential of MSCs. Janani is the one who works with me, and she's the one who is responsible for making all 3D bioprinted, biofabricated ones. So while I move towards an end, I have last 2 minutes. So it's a remarkable promise, and the expectations don't have much of a gulf. It's really a reality. I believe the data, a little bit what I could share, must have been making you understand that we need to hasten a bit through various biotechnological interventions. I shall fail in my duty if I do not mention IVRI. I come from the first batch of PhD from IVRI, and this is an institute where I developed majority of the data. This is the acknowledgements which are given to Team IVRI, embryonic adult fetal stem cells, Dr. Amarpal and his team, a whole team of OPD and surgery, and Dr. Rath, till I did not have my own in vivo imager, used to go to CDRI to work with it. Dr. Sai Kumar to make every pink and blue pictures of histopathology. Dr. Bhaskar Sharma, I was using majority of the facts, work with him, and all the funding agencies to give crores to us and believing in our science and flushing the funds to us so we could do a little bit whatever we could understand. And of course, my own institute, NIAB, which is one of the best place in the country for the research. I'm not biased, but the truth. And I like to thank all of you. Hima, I suppose I could respect the time. And I would really like to say in the end, thank you to all of you. And I would like to say, please don't follow anyone. Differentiate yourself, what you are from others. With that, I close my talk, and I'll really be very happy to take any questions if I could.
Thank you, ma'am. Thank you so much.
Thank you very much, ma'am.
Thank you very much.
Thank you very much for your
Hima, can
talk.
my screen be unshared now?
Yes.
I stopped sharing. Thank you. Thank you very much.
We have some questions from the chat.
Yes, we have received one question in the chat, and also I request all the participants, I'm sure you would have comments, questions, or doubts related to the result. So I think I see one hand up, so I would unmute Mudashir Bashir now for asking the question. Please go ahead. You are muted, Mr. Mudashir. Okay. Till then, we'll bring out a question from the question box. So we have one question from Bhuvan Tripathi, who has asked that as we know that stem cells are grown in the laboratory or adult cells are reprogrammed to become stem cells, there might be genetic damage. How researchers overcome this problem? So Bhuvan, if you are there and you would like to ask the question yourself, please. Otherwise, I will pass on the question to Dr. Sharma.
Okay. Okay. Thank you for that question, Bhuvan. And I believe he's talking about the iPSCs, because when he says that we convert, so the point of genetic mutations or genetic defects come when we are compromising with the cell types. Here, we would not like to do that. Whenever we are going for generation of iPSCs, one would be very critical and careful taking a type of fibroblast or type of somatic cells, which we would like to get them converted towards iPSCs using a Yamanaka factor. It's very simple. Bhuvan, are you there? I suppose I could. Mudashir?
Okay, next we have.
Good morning to all. Actually, I don't have a question. Actually, I'm really thankful to ma'am for giving us the opportunity to be taught to learn from the ma'am who has done a lot of work over here. Actually, what I was doing over in that, boy, I raised hand, ma'am told that we have a problem with respect to veterinary science, why we don't have the guidelines related to stem cell therapy in the veterinary sector. For that matter, ma'am told that you may raise your hands that there is a problem and we need to have a solution to it. That's why I was raised my hand at that time. Otherwise, I don't have a question right now related to any problem. Thank you so much, ma'am, for being there.
Thank you, Mudashir.
Thank you.
Next, I can see Vimal Anuj raising the hand. So I'll unmute Anuj. Can you speak?
Yeah, am I audible?
Yes, yes.
Yeah, Thank
Yes,
you, and
please.
thank you to Dr. Sharma for that enlightening lecture. And I just want to know that in the last 2 to 3 slides, you are mentioning that you are just planning to go for this kind of guidelines and all that. So I just want to know that will it the CRISPR technology will also be considered for the new set of guidelines while going for the stem cell research? And is there any lab in our country which is currently working on this CRISPR with related to stem cell therapy and all that?
Okay, thank you for that question. So the guidelines right now, we're just not focusing on CRISPR. Yeah, there are laboratories which are focusing one or two, and those laboratories are not using the CRISPR technology to use these cells towards a therapeutic application. They are mainly using a CRISPR technology, especially when it comes on a iPSCs or iPS related work, and mainly in the basic biology group they are doing in generating that data. So right now what our guidelines are, which we are focusing is on, it's actually very simple, whether we could use the stem cells of animal origin in different animals or livestock species for therapy, yes or no, is a major focus of a guideline. However, the CRISPR work would eventually get covered if we talk about iPSCs, which is a part of ICMR DBT guidelines.
Yeah, thank you. And because stem cell work to do this kind of work is really because a lot of ethics are involved and bioethical issues are
Yes,
there. So
yes.
that's why.
Oh, sure. Oh, sure. And moreover, whatever work we do, anything, everything, animals, as all of us know, you can do with the IAEC work or Institute Committee for Stem Cell Research and Therapy. These two committees can do. But the moment you go and touch either canine or caprine or livestock, you just cannot do it unless you have CPCSA clearance, right? So guidelines are still there. However, the guidelines to use the stem cells in these animals are a limitation.
Now, next I can see Vimal Singh with his hand up. So please, I will be unmuting. Please go ahead with your question. Vimal Singh, can you hear me?
Hello.
Hello, yes, you had a question. So if you'd
Yes.
like to ask it yourself.
Yeah. Can you hear me?
Yes.
Okay. Good morning, everyone. That was a fantastic talk, and a lot working in the same field for the last 10-15 years. I would
We
like to
missed
know
you.
what is the most clinically acceptable feeder-free methodology we are having in Indian scenario.
So I like to tell you, the feeder culture is only required when you go for either the embryonic stem cells or the iPSCs. Otherwise,
Yeah, that's what I'm talking.
Otherwise, the moment you take adult stem cells or fetal stem cells, they are the surface charge, they have surface charges, so they get attached onto plastics. So that's the advantage of using the adult and fetal stem cells.
I mean, clinically acceptable methodology. Do we have any standardized method SOP for clinical acceptance, or still in the research phase? Is there any existing example in any hospital or in any scenario who has used those kind of iPSC derived finally therapeutic?
In case of animals?
In case of human, clinical acceptance.
In case of human, in case of human, iPSC cells makes more of a sense. Reason being, you are neither using embryos and they are also not the ones which will form teratoma. Fine. So there is a better use compared to the animal veterinary sector. One. But clinically, people are not using it much because there are better and easier and cheaper and simpler methods available. For example, adipose origin stem cells, adult origin stem cells, placental origin stem cells, and manseral origin stem cells, right?
But all those cells, they also have their own limitations. They do not have the potential to give you the final product what we want for the therapeutic purpose. That's why iPSC were initially launched. And now I do understand that what tissue specific cell can also be a good starting point. And okay, I understood. The second point is, do we have any Indian platform where we can get all these cell lines? Because NCCS recently had their own website that they will be making it available soon, but still there is no availability, and all these
Yes,
cell lines are
yes.
very expensive to get from outside.
NCCS does not have these cell lines, you're right. And if you want these cell lines, we have at IVRI and we do have at NIAB, though we have to make them officially available for others. This is the current status. I am talking about the animal origin stem cells.
Okay, so
And
they are available with
Ratan
the
wanted
price
to add
or
up something.
sharing base? So anybody can purchase them, or do we need to have a collaboration, or do we need a grant?
Well, right now we don't have a purchase mechanism. We only have a collaborative mechanism. May I know where are you housed?
Presently, I'm working in MIT University, Haryana. Earlier, I was working in Delhi Technological University and DRDO in Mass. So I have
So what I suggest you, if you have a keenness to work, please do approach us, and we would really like to share the cells which we have with us, the adult ones. We don't have the embryonic with us, adult ones. I believe Dr. say something. Thank you. Dr. Ratan wanted to say something.
I will write to you in order to get more detail. Okay, thank you very much.
Sure. Most welcome.
Yeah, ma'am, I want to add one point here as a response to the last person who asked the question. Using the iPSCs in therapeutic, promises, one issue is that it works on all and none function, So once you take the iPSCs and tell them to differentiate, everyone will differentiate. Okay, or they totally fail to differentiate. Wherever the madam is working on the adult stem cells, tissue-specific stem cells, now they divide in a asymmetrical fashion, and only those cells or the amount of or the number of cells required for the repair will be differentiated, and they'll maintain the stem cell pool. So probably I think that could be a possible reason that the therapeutic potential of tissue-specific stem cell is gaining popularity. Thank you.
Yeah, I think that's a brilliant point you made. Thank you very much, Dr. Ratan. So I believe we could make it together clear to you, and you're most welcome to approach us. We would like to share. We would not hesitate sharing cells with you, what we have.
Okay, thank you.
Any other questions?
Yes, we have 2 more hands up. So I think a lot of people have benefited from the last answer, and they would be approaching you soon for more information.
Oh, sure.
So next I can see Teja and Swati. So I'm going to unmute Swati for now. Please go ahead and ask your question.
Hello, good morning, everyone. And good morning, ma'am.
Hi, Swati.
After so long. How are you, ma'am?
Good, fine. How is yours doing?
I'm good. I'm good too, ma'am. And thank you for letting me know. Like this webinar is happening. It was nice to know. Like you elaborated it so well. Like you connected both.
Thank you.
You used both clinical and the basic science in a very beautiful way. So my question to you was like, what are the funding opportunities for the young researchers in India, because stem cell work is pretty expensive, and belong to a developing country, like what are the funding opportunities would be for researchers like me if we will be coming back?
Thank you very much, Swati, for this good question. So you have a very important person amongst us, Hema. They are going to do some advertisement is soon going to come for the clinical research. If I'm correct, Hema, it's there on the website is one. And Swati, for people like you who have been trained here in IIT and IVRI and who are sitting in US and you like to come back, Ramalinga Swami Re-Entry Fellowship is already advertised. You do apply. You got a Ramanujam Re-Entry Fellow, you can apply. There is a Wise Kiran and DST has already given. Dr. Gupta has already quoted in Twitter, and the list is long, and they so brilliantly have detailed out what are the funding agency, and you could do it. So ample opportunities to come back.
Okay, thank you, ma'am. Thank you so much.
Thank you.
Thank you for the question. And next we move on to, I think we have almost covered. I move on to Teja now. I'm unmuting you. Can you ask a question, Teja? Yeah, sure.
Yeah, ma'am. Are you able to hear me now?
Yeah,
Yes.
Yeah. Thank you very much for the opportunity, ma'am. It was a very enlightening session. So, ma'am, I am basically a clinician, MD, it's general medicine. So, my question is, like, how far are we away from using the stem cells for clinical application in human beings? So, all the things that we have discussed, whether the fractures, spinal cord injury, etc., we see a huge potential to be used in human beings and for the benefit of entire mankind. So, what is the present state of stem cell research or conditioned medium application in India, and what are the regulators that we had to clear through before getting
okay.
into the market or using it on the patients? Thank
Yeah.
you, ma'am.
Thank you for that question, Teja. Wonderful. So it's not only opportunities. Opportunities have been happening last more than 8-10 years. AIMS, you have got a special cell. It's called Orbo. And Orbo people, we have a stem cell biologist and a stem cell group, and they have
Okay.
a kind of collaborative work which they do with the very many clinicians like you. Where are you housed, Teja, if I may know?
Currently, I'm working in a private hospital in Hyderabad.
Okay, no
So,
problem. So I
just
I
in
can
clinical
get you
application.
connected with the right people who are doing,
Okay,
and
ma'am.
in New Delhi they are doing it from last more than a decade or so, and
Okay.
especially the bone marrow transplant is
Okay, ma'am.
very successfully done. Salutes to those clinicians who are doing it. One of my own colleague has gone through it, and Touchwood is doing very well. So it's really
Great.
happening. It's really happening. And also, if you are aware, the country already has the stem cell collection and stem cell banks once you have a child. See, when my children were born, this facility was not there, so I could never, preserve the cord blood stem cells. But right now, it's available, and it's as economic as less than a cost of a mobile, hardly 19,000, 20,000. So it's
Okay.
all happening. It's all happening. It's a reality.
No.
Yes.
And application of conditioned medium use as of now in human beings, ma'am.
No, no, that's not happening. So that there's a lot of opportunities. Maybe nobody's doing. I don't know how young you are. You can think of starting it in case of human beings. It's not happening in human beings.
Right, ma'am. Okay. Thank you for Thank you, ma'am.
Thank you. We cannot hear you,
Thank you for the,
Hima.
yeah, thank you for the question. And I can see a lot of hands up and questions up there. So I don't know how much we can take, but next we have Mamoni Das. So I will unmute her for her question, and then we'll see if we can manage one to squeeze in one more. Mamoni, I have unmuted.
Good morning, everyone. Am I audible now?
Yes.
Madam, namaskar.
Namaskar, Mamani. How are you?
I'm good, ma'am. Very nice talk, ma'am. From going to the simple concepts to all your fantastic work that has already been validated in veterinary sciences. So it was a very good overview that I could myself have as well. So my
Thank you.
question is more related to the material aspect. So I was wondering, when you are making the scaffolds for the challenge that you put in the bone tissue engineering segment, where one of the issues was the mechanical integrity. So what are the approaches that you think are most like promising now in veterinary sciences,
Okay.
especially?
Okay. So other than yeah, thank you very much. This is a wonderful question, very important question as well. So other than the materials, yeah, material science has grown enough and we have very many good groups doing it. But the important part here is when we go for this, especially the bone aspect part of it, people who are culturing the osteoclast and osteoblast like you and coming forward and getting it inserted along with would surely make this a more robust thing. It's that simple.
Yes, ma'am, because I already see that you have moved to electrospinning materials, and so you might be having now a strong hold on how you can change the mechanical properties if you get into the micro and the nano level. So,
Yes, we do. We do.
yes, so I do understand what you are indicating at, like the co-culture models and all might be very, very helpful. Thank you very
Yeah.
much once again, ma'am,
Thank
and
you.
I hope to
Actually,
get
this credit
connected
goes
soon.
to the wonderful scientists out here who are
Yes.
great in making those electrospun nanofibers and the ones who are good at making 3D biofabricated devices because it's their area and I just
Yeah.
say we need to scaffold and that's it. I'm basically a
Yes.
strength one scaffolding and
Yes.
getting help
Yes.
from wonderful people. Thank you.
Yes. Yes. Thank you, ma'am.
Thank you so much. Now we have one more person and we'll take one question. Sadish has been putting a lot of questions on the chat box, so I thought, if we, if you can unmute himself and unmute and speak. Are you there? So I think Sadish does not have the option of asking, so I'll just share the question with Dr. Sharma.
Yeah, sure.
Dr. Sharma, can you see the question?
No, I cannot.
Okay, so I'll read it out. So what he has said is, thank you for giving an excellent presentation. The healing properties of stem cell mitochondria are also a factor, as you mentioned here. Since the mitochondrial phenotype is different among individuals, how mitochondria is involved here? I am.
Who's that asking question, please? Can you let me know?
Sandesh, I have unmuted. So can you, if you could elaborate on the question,
Very good
Sandesh?
question. Very good question. What's the name you said?
The name is given as Sadish EM.
Okay. Hello, can you hear me?
Yes.
Right. Anyway, I can answer. So that's a very good question. Very, very good question. And my answer is no. We haven't done any such work wherein we could ever relate to mitochondrial DNA details. But yes, it's a typically parsed area and one can really think of. Well, I don't have any data, but I go with you physiologically. Yes, it does. It is different. But my answer to this is no, no data with me. Thank you.
Thank you everyone for the wonderful questions that brought out a lot of, clarified a lot of things for the rest of us too. And a special thanks to Dr. Taru Sharma for the wonderful talk today. She started from the basics and took us through the different phases, talking about the clinical aspects. So we are really thankful to have her and grateful to have her as a speaker today. And also thank you, ma'am, for the giving the opportunity to share your own work and everything with the participants. So we judging by the number of questions and the interest the participants have shown, I'm sure they have benefited a lot from the topic.
I'm still holding my questions, so maybe ask sometime later.
Please go ahead. It will benefit everyone.
Sure. Dr. Ratan, please.
Short as a question, ma'am. I don't know if we have a specific answer to that. My question was where India should focus on the stem cell research in next 10 years, and how you see that whether the application will come into the practice in veterinary and animal sciences. How you see to it, ma'am?
Yeah, wonderful question, and again a next brilliant one. So what I personally feel that with so much so we have moved ahead, we need to focus mainly on the large bone fracture if it comes on a livestock sector, for sure. That's one. And another crucial thing is all the memory origin stem cells and the area wherein you are there and working to address the mastitis, which is a multi-etiological problem. And it's not so easy to do, but yes, that is another one. People are approaching through different manners, AMR approach, antibacterial approach, antimicrobial approach, and so much so. But this is one approach which is really to be taken ahead. And if you say a decade, and within a decade, if we are able to address large bone fractures, I'm specifying and repeating again, should be our focus for sure. And also, along with this, it could be made doable if we could have, someone has asked a very good question from MET, or do we have a stem cell banks? So we don't have. So we need to have within next, I'm restlessly at it, the stem cell banks, so that everyone need not to make cells, there should be specified at a national repository wherein people could just, like NCCS is having, some cells. So we could also think of having a specific animal stem cell banks. It's a requirement. These two things are the most priority in my list. Thank you.
Thank you, ma'am. That answers my question. Thank you so much, ma'am.
Yeah, thank you very much. Yeah. So what Hema was saying, so I believe the whole credit goes to Dr. Malik and Dr. Ratan for having me here and giving me opportunity to share little bit whatever we could do both at IVRI and NIAB. With all humility, I really appreciate, and Hema has been pretty kind to do so. Yeah. Thank you.
Yeah.
Thank you. So we did get a few questions on funding opportunities at India Alliance. And you can see my colleague Isha Goel ready with her presentation now. So let me briefly introduce you to her. Isha is a nanobiotechnology engineer possessing an MTech in nanobiotechnology from IIT Roorkee and BTech in biotechnology from MIT Noida. She has an ardent research experience, especially in the biomedical domain, which has translated into a patent for burn wound healing patch. She has worked as a project executive in biotechnology firms in Delhi, where she was part of the management team for National Plant Tissue Culture Certification System, training program for African candidates, and setup of new ventures. She's currently a grants advisor at India Alliance and the best person to talk about funding opportunities at India Alliance. So over to you, Isha.
Thank you, Himashri. I'll share my screen now. Are my slides visible? Thank you. So, good morning, everyone. Thank you for the warm introduction, Imashri. Today, I will be talking about the various funding opportunities that India Alliance offers that you can leverage and help give you a snapshot of all the opportunity that India Alliance brings to you all. India Alliance is a public charity that funds research in health and biomedical sciences in India. We are funded by Department of Biotechnology, Government of India and the Wellcome Trust UK, and we invest in transformative ideas and supportive research ecosystems to advance discovery and innovation to improve health and well-being. This is our mandate. So, since the launch of India Alliance in 2008, we have successfully steered fellowship programs in both biomedical research, started in 2008, and research in clinical and public health area started in 2013. In our second phase, which was started in 2019, India Alliance launched a new grant programs, new promoting clinical and public health research centers and team science grants, which are collaborative high-risk research grants. This slide shows the pan-India reach of India Alliance since 2009. We have given out 550 awards in across 137 organizations in 47 cities across India. And this map is being filled rapidly every year. If we have to look by the types of institutions that have received funding from India Alliance, you can see that we have funded institutions from academic universities to academic research institutes to private universities, academic research institutes, central universities. All institutions have been a part of our research funding portfolio. India Alliance has an arsenal of innovative funding schemes and aiming to build research capacity in India and catalyze internationally competitive research. I'll first briefly take you through the features of our flagship fellowship schemes, and then we will move to the grant schemes. India Alliance fellowship programs have very attractive built-in features. Here, like these set us apart from the other fellowships that are there. Like at India Alliance, more emphasis is on the lead investigator. There is no bar on age or nationality. We give due regard to non-research career breaks provided they are properly justified in the applications. So a break in research, for example, due to unprecedented pandemic or maternity reasons is given due regard. A salaried position at host institution is not a requirement. We have an online user-friendly application process through the ISS portal. We uh, we encourage fellows to pursue interdisciplinary research, and our awardees have provisions to develop international collaborations. Selected applications go through international peer review, where applicants receive insights and suggestions on their proposals. The most important feature of India Alliance funding is the quanta of funding is generous, and the fellows have a lot of flexibilities in how they utilize their funds. Our fellowship programs provide funding for individual scientists at different stages in their careers. Like early career fellowships are designed for talented, newly qualified PhDs and postdoctoral researchers to attain independence. Intermediate fellowships are for researchers who wish to establish an independent laboratory. Uh, Another thing of intermediate fellowships that is seen is that it has achieved to bring researchers from abroad to the Indian scientific research fold by providing generous and flexible funding. Senior fellowships are for more established researchers who have demonstrated their ability to lead an independent research project and group and want to expand their research program in India. All these three fellowships are available for both the basic biomedical researchers as well as for clinicians and public health workers. You may access information related to all schemes on our official website. Here, I would like to briefly go through all the key provisions of our fellowships. So, starting with the basic biomedical fellowship schemes, for early career fellowship, it is a mentored fellowship. A point to note is that to foster independence, early career fellows are strongly encouraged to carry out their fellowship project in a laboratory that is different from where they have completed their thesis. Eligibility for early career fellowship is from the last year of their PhD to 4 years post PhD or an equivalent degree. This competition occurs once every year. Senior and intermediate fellowship, that is the SIF competition. This competition is open to researchers with 4 to 15 years post PhD or equivalent degree research experience. Applicants can choose the scheme most appropriate for them based on their qualification and career trajectory. The senior and intermediate competition runs twice a year. As I said earlier, these are very generous fellowships and have a budget cap of 1.7 crore for early, 3.6 crores for intermediate, and 4.5 crores for senior for the duration of 5 years. Salary, it is included in the budget. Fellows are provided with salary support set by India Alliance for each scheme. In case a fellow receives a salary from their host institution, a top-up is provided so as to meet the salary level set for each category by the India Alliance. The fellowship can also be used to purchase equipment through funds requested, though funds requested for expensive equipment are typically not allowed for early career fellows. It is expected that applicants will choose appropriate laboratories or environments where majority of the necessary equipment and facilities are already available. For intermediate and senior fellows, there is no such restriction. There is also the provision of hiring support staff. ECF can request for only one technical support staff, whereas intermediate can take up to 2 and senior fellow can take up to 4 support staffs, including PhD students and postdocs. WOHI, like WOHI is work outside host institution. Like mentioned earlier, one of the coveted provisions of our fellowship is that fellows can carry out a part of their work at different lab outside their host institution. This can be in India or abroad. For working abroad, fellows will be provided with an allowance of $3,000 per month based on the prevailing conversion rate. Early career fellows can visit their a lab outside their host institution for 2 years, while intermediate fellows can avail WOHI for a maximum of 1 year. The WOHI option is not available for senior fellows, but they may request for funds for travel and subsistence costs for short visits to collaborators. In addition, for travel to conference, meetings, or visit to collaborators' lab, we provide up to 7.5 lakhs for 5 years for early and intermediate, and up to 10 lakhs for 5 years is provided for senior fellows. Finally, contingency funds are provided as India Alliance recognizes that unanticipated costs might arise during the tenure of the project. Therefore, in addition to the requested costs, the India Alliance will provide contingency funds to cover such expenses. Moving on to the clinical and public health schemes. So, an overview, I'll quickly point the main differences between the clinical and public health scheme as compared to the basic biomedical schemes that we have discussed just now. Of course, the eligibility criteria is broadened to include MD, Masters in Public Health, and PhDs for the clinical and public health fellowship schemes. As clinicians and public health researchers often have very different career trajectories compared to basic biomedical scientists, our eligibility also takes into consideration and is more flexible in terms of length of research experience, which you can see is from last years of PhD to 15 years post PhD or equivalent degree. Applicants can choose the scheme most appropriate for them based on their qualification, research experience, career trajectory, and track record. The salary cap for and budget cap is also slightly higher for clinical and public health researchers, mainly to compensate their clinical practice. Since many clinical and public health schemes also require field workers, etc., the provision for support staff is also relaxed, subject to strong justification. Other provisions remain the same. Here is a representation of the number of areas of biomedical and clinical and public health sciences that are getting covered through India Alliance fellowships and grants. You can see the broad remit of India Alliance Fellowship Scheme is to support biomedical research that is relevant to human and animal welfare. Within this broad remit, our fellows are from a variety of fields ranging from molecular biology to infectious diseases, structural biology, bioinformatics to public health. This layout is a representation of the various areas of our funding. Coming to the application process to become an India Alliance Fellow. All our fellowship schemes require a preliminary application, which includes the applicant's CV, a 750-word abstract, and at least one recommendation letter. Based on this prelim application, we assess the eligibility and competitiveness in the cohort, and then the full applications are invited. Full applications are accepted by invitation only. The full application is a more comprehensive form with multiple sections that an applicant needs to fill out. Like it has more detailed proposal, requires letters from all roles of your application. It has a detailed budget section, etc. And then these full applications are sent out for international peer review, which is an important step of our application cycle. The review feedback is shared with all applicants at the end of the regardless of the shortlisting decision to help craft their future proposals. Based on the review feedback, applications are shortlisted for interview by the selection committee. Then based on the assessment at the interview, applicants are chosen for award or declined. This entire cycle takes about 10 months to complete. An important thing to note here is that once you submit a full application, you cannot compete again in the same competition or a junior competition. So if you have submitted a full application for intermediate fellowship, you cannot apply again for early or intermediate fellowship regardless of the decision. You can, however, apply for a senior fellowship. This is the India Alliance resubmission policy, which I wanted to highlight here. So keep an eye out for call for applications on our website and social media handles and check eligibility and submit the application through the online portal management system ISIS. This is a snapshot of our online portal for applications. So what makes an application strong? Uh, it it takes a strong motivation. You need to show scientific productivity in the form of publications, mentoring, and outreach events that you take part in. You need to present a feasible research plan answering an important biomedical question, demonstrating expertise and ownership of the idea, good grantsmanship in answering all questions in the application in detail, supportive letters of support from all roles in your application, like collaborators, mentors, supervisors, willingness to move out of comfort zone, and justifying all your choices you have made and shown in your application. Having covered fellowships at India Alliance, we'll be moving on to the grants funding programs India Alliance has launched. I will now briefly talk about the team science grants and the clinical and public health research centers. The team science grants will fund team of researchers who bring together complementary skills, knowledge and resources to address an important health challenge for India. These grants also fund high-risk, high-reward research work. The project should be multi-institutional and interdisciplinary in its approach. A minimum of three investigators should participate, including one as a principal investigator who would manage and lead the team. The applying team should bring different expertise or discipline to address the research problem within the remit of India Alliance. No more than two investigators can be from one institution, and a minimum of two institutions should be included. We encourage to observe gender balance and geographical diversity in the team. The funding provided is up to 10 crores for a 5-year project where funds can be requested for staff, equipment, consumables, strengthening research management structure at the institution, etc. Moving on to the clinical and public health research centers, are institutional grants that are meant to promote clinical and public health research and develop physician scientists. They would empower clinicians and public health scientists to carry out patient and population oriented research in addition to patient care and preventive medicine. In short, these are called CRCs. The CRCs are envisioned to include more than one institution. Strong justification would be required for a single institution CRC application, in which case multiple departments at the institution should come together to make that application. Here also the funding provided is up to 10 crores for a 5-year project. Funds can be requested for hiring equipment, maintaining clinical and population cohorts, training early career clinicians in research, strengthen research management structures, etc. This is Clinical Research Training Program, CRTP. The, this is clear aims. This aims to develop physician scientists as a way uh, forward for improving clinical and public health research ecosystems in India. CRTP is not a standalone program and will be administered embedded within the CRC program, which was discussed in the previous slide. The program would fund 3 to 4 year mentored research training fellowships for medical graduates, MBBS, and postgraduates, MD, MS, and equivalent. Up to 12 fellowships will be available each year, and the value of each fellowship will be INR 50 lakhs. Only personal support for the fellow is provided in the CRTP scheme. This is our TSG CRC footprint, uh, which network, which is increasing every year. We launched our new funding scheme India Research Management Initiative. With the this is a new scheme for research managers, and it has the purpose of creating robust research ecosystem in India to address global challenges. IRMI, in short, for India Research Management Initiative, aims to strengthen institutional ecosystems by facilitating setting up of research management systems through funding, training, and mentoring. Here, so in addition to identifying and supporting the best scientific talent in India, India Alliance is also trying to create a robust network of research managers across India. The India Research Management Initiative began in 2018 and is currently successfully creating a community of research managers. IRMI offers funding opportunities for research managers. There are three types of competitions. First, to launch a research manager's career is a 3-year research management fellowship. Second, to support existing research management offices through a 1-year IRMI grant. And third, provides networking opportunities through a 6 months IRMI travel grant, which they use to travel internationally to research management conferences and meetings. These maps show the institutions who have received research management grants and fellowships. Every year, new institutions are being added to the fold. Those who are interested can join the IRMI network by submitting the individual and institutional registration form from our website. As you can see, the IRMI network encompasses individuals across the length and breadth of our country. This is not all. There are more India Alliance initiatives to support and empower researchers. Like to encourage resource and knowledge sharing, India Alliance, in collaboration with EMBO, European Molecular Biology Organization, launches lecture series every year to teach contemporary topics in life sciences. Also, lab leadership workshops are launched to cultivate leadership skills and develop management skills in our researchers. Also, additionally, research methodology workshops are organized, providing structured training in epidemiological study design, data collection and analysis, and for developing Indian physician scientists, DIPS workshop, which is developing Indian physician scientist workshop, is organized to ignite scientific curiosity in young doctors. India Alliance aims to make science accessible to all through the open access publication. India Alliance has joined the Wellcome Open Trust and Europe PMC to ensure adoption of its open access policy. India Alliance organizes various and supports various public engagement, scientific communication events all year round. India Alliance has trained more than 9,000 researchers from over 120 Indian institutions through its science communication workshops, and India Alliance runs and funds various public engagement programs which bring the scientific community and public together on one platform. Some examples of India Alliance public engagement programs are the early years, a window of opportunity, which is run with programs like which are run in partnership with various universities. Public engagement events are held at schools and in partnership with various organizations to create more awareness about the science in India. India Alliance has its in-house media and publishing, where our external newsletter brings to you the latest updates and funding opportunities at India Alliance. Our videos and podcast series highlight the impact our research fellows and grantees are making through their research in society. India Alliance public engagement seed funding has supported um, Written Out of History, Forgotten Indian Scientists, a book, and various events like plenary, like even going as to the far corners of Arunachal Pradesh to create more public engagement, and podcasts like Schools and Leaves. With this, I come to an end of my presentation and would like to draw everyone's attention to the upcoming competitions. Our tentative competition timelines are mentioned here. All fellowship and grant competitions will open sequentially in 2023, with TSG CRC competition to be launched soon. Grantees to know more about our various funding schemes, do look up on our website and follow our social media handles. You will find a lot of interesting stuff there. Write to us at info@indiaalliance.org if you have any doubts about your eligibility or scheme provisions. We would be happy to help. With this, I would like to close my talk. Thank you for your time and attention.
Thank you, Isha, for the detailed information about India Alliance, various opportunities at India Alliance. And if there are any questions from the audience about any of the grants or programs, please feel free to ask the questions. Raise your hand and ask the questions. Also, if you want to apply, go look to our website, and while applying, you have any issues, then please write to the email ID that Isha has shared about info@indiaalliance.org, so, and they will answer the inquiry 3 to 4 days. And I will also ask Dr. Sharma or Dr. Chaudhary to let ask if there are any queries from your side.
No, I think this was wonderful. Isha has really detailed it out. So we knew few, but
I cannot hear you.
Okay. I can hear you.
Okay.
Dr. Taru, you were saying something.
yeah, I was just saying that you could make things much more easier. We were knowing about a couple of it, but you, especially me and all, which you have just said, that's fantastic. That I was not aware about. And I think I was already talking to Dr. Himashri yesterday, and I'll be reaching out to you with the more specific, which is there. I really want to talk one-on-one. Thank you very much for updating it.
Sure, Dr. Taru, we are most welcome, and we'll any doubts or any interest you have in any of our schemes, we can surely guide you.
See one hand up, Teja. So I'll unmute you.
Yeah, thank you for the opportunity again, ma'am. So my question is like, so I am an independent individual private practitioner based in Hyderabad. So can I, and I have few like research fellowships during my MD and MBBS periods in the basic science as well. So now can I apply as an independent or individual practitioner for these fellowships and then associate with a lab over there with a central lab also. Is it possible?
Okay. From what I understand, there's one point that I would like to highlight. In like the institution that you are associated with needs to be a not-for-profit organization. So,
Okay.
following if like where you are practicing, it's a not-for-profit organization, then India Alliance you are eligible to apply for India Alliance funding. Does this
Right.
help you?
No. Yes, yes, yes. Absolutely. Yeah. Thank you
Okay.
so much.
Thank you.
Thank you.
I see Anuj with another question. So I'll unmute Anuj. Please go ahead.
Am I audible?
Yes.
Yes.
Yeah. Thank you for the opportunity once again. So I just want to know that is there any sort of help or mentorship like opportunity if we are going for any application so that we can have some sort of guidance or like that input insights prior to application from India Alliance?
Sure. In like, if you have any queries, you can write to us before even making an application to India Alliance. You can write to us regarding if you want to know your eligibility or which is the best scheme for you to apply to. And surely you can write at info@India Alliance, and our grant advisors will guide you through the best. And also India Alliance organizes various webinars where we go section by section through the application and guide applicants in how to fill the form, what to prioritize in each section, and these webinars are also available on YouTube. So,
Okay.
and as and when a competition is launched, prior to that, this webinar is organized before every competition. So that also helps our applicants to write, put their best foot forward in every competition. So you
Okay.
can mainly follow our social handles, and soon updates for these will be showing up there.
Thanks to Dr. Huma, Dr. Sharma, and this final session. A huge round of applause for all of you. A huge round of applause.
Thank you.
Thank
We
you.
highly appreciate your appreciation.
So thank you. If there are any more questions, please feel free to ask. And you can always, like Isha mentioned, our social media handles are very active. And as you could probably be able to see right now, also we have posted something that which is coming soon. We have our team science grants and clinical public health research center grants coming soon. So you can begin your preparations by going through all the YouTube videos that are uploaded last year, and go through the eligibility and application process before we launch it so that you are prepared. So now if there are no more questions, then we will move to the final section of today's topics. So I would now request Dr. Ratan Chaudhary again to give a vote of thanks for the entire series. So over to you, Dr. Chaudhary.
Okay, thank you, Hima. Dr. Malik has gone for a meeting, so he's not available. I'm sorry for that. But on behalf of GADVASU and IIT Roorkee, I, Dr. Ratan Kumar Chaudhary, Assistant Professor of the College of Animal Biotechnology, Guru Angad Dev Veterinary and Animal Sciences University, Ludhiana, Punjab, feel privileged to propose a vote of thanks for the sixth lecture of Continue Biotechnology Education, that is a CBE lecture series. First of all, special thanks go to our eminent speaker and dignified researcher of international repute, Dr. Taru Sharma, the director of NIAB Hyderabad, for providing basic and advanced information about the stem cells. She enlightens the importance of stem cell in therapy of the animal diseases and showed convincing data in treating incurable disease of canines like problematic wounds, hip dysplasia, spinal cord injury. She also showed other areas where stem cell find its applications like tissue remodeling, understanding early embryonic development, drug testing, cell-based therapy, and many more. So thank you so much, ma'am, for accepting our invitation and enlightening us about these stem cells. Thank you so much, ma'am. I also thank the worthy Vice Chancellor of GADVASU, Dr. Inderjeet Singh, who has constantly been there to support, encourage, and guide us. Thank you very much, sir, for your envision, encouragement, and recognizing the importance of biotechnology in unfolding the problems of human and animal health, and hence allowing us to conduct such relevant lecture series. I express my cordial and sincere gratitude to our head of the family, the Dean, and the coordinator of CBE program, Dr. Yashpal Singh Malik, for all technical help, moral support, and guidance. Thank you, sir. I would like to thank Himashri Bhattacharya of India Alliance, the lady being organizing, hosting, and managing today's event. I would also like to thank Isha Goel for providing an overview of funding opportunities at India Alliance. Thank you so much. Ultimately, and I thank all the participants who have shown keen interest in this program and participated in the CBE lecture series. Please provide your feedback and once you receive a certificate from the DBT. Once again, thank you so much. Stay safe and healthy and have a great day ahead. And I'm hoping to have a more prospective collaboration with the DBTA to a greater extent and for a longer duration in coming years. So thank you so much.
Thank you, Dr. Ratan, for the wonderful end to the day. So I just have an important announcement before we leave for the day. Once this webinar session ends, you will be directed for a quick survey. The participants will be directed to that. So please take a minute to provide your valuable feedback. It will help improve our deliverables in future. And also, I take this opportunity to thank our partners, the management of GADVASU, Dr. Yashpal Malik, Dr. Ratan Chaudhary, and IIT Roorkee team for their guidance and support in making this endeavor a success. And I also thank all the speakers of the past webinars also. And if any of you missed the earlier webinars, you can listen to them by going to the India Alliance YouTube channel. And of course, to keep following us for more such events in future. And I hope we'll be back soon with another interesting series of lectures. So thanks to all the speakers of the day.
I think please share the link of the YouTube to the madam so that she can put on her website. It will be nice for others who couldn't listen today's lecture. It's very important.
Sure. Thank you. Thank you. It was nice.
Thank you very much. Get connected.
Thank you.